So the next thing we have to talk about is the method of development of the class. And you're like, huh? And then we have, how's the gastrula formed? The first thing we have to think about is, what does the term gastrula mean? This term gastrula basically means stomach. And as you look at the stomach or digestive, you're looking at how does that digestive system, how does that method of absorption form. We learned in the video that there's three main germ layers: an ectoderm, an endoderm, and a mesoderm. And we actually even learned some of the parts of a human that are made up of those. But as we look at the mesoderm, actually doesn't always form. And it's the last layer to fully form. So as you look at advanced organisms, they are going to form things like their brain and other really important tissues from that mesoderm. But more simple organisms may not have as advanced of a mesoderm. So the next thing we look at is where does the mouth opening? So does it have a complete gastrula? Where does the mouth opening when that blastula folds in? And we have a protostome and a deuterostome. What I'm talking about in this is, so if I have my hollow ball of cells, okay, so each this outer line would be made up of a ring of cells, okay? And basically what happens is this ring of cells pinch in. So it actually kind of pinches downward. And then as those tissues form, this becomes the endoderm, and this becomes the ectoderm. And so we have this gray area of where does that mesoderm actually form. So as you look at that, we're also looking at not only is there a mesoderm, but there's also an opening that forms. So if it deforms directly from the outside of this, that becomes a protostome. If it pinches off and folds outside over here, it becomes a deuterosome. Okay, so those are the two different formations of the gastrula. Does the mouth become at the end, or does it form from somewhere else? The other thing we have is a ceolomate cavity, and we're going to go as we look at how this folds in and the three different layers really form. So as you look at a germ layer, you have three main layers. You have your endoderm, which is going to be on the inside, your ectoderm, which is on the outside, and then you also have a mesoderm. Okay? Different cavities within the body. So as we look at those cavities, we basically have three different types. And this mainly becomes in organisms with bilateral symmetry. Other organisms tend to have either part of those or not those at all. So as we look at those, we have three major different cavity-filled spaces. We have an acoelomate, so coelom is going to be the fluid-filled space. We have a pseudocoelomate, and we have a coelomate. So we know that A means without, pseudo means false, and then you have one with that actual fluid-filled cavity. So as you look at this, you can kind of categorize these according to the organism. So an acoelomate is going to be something like a flatworm, a roundworm is going to have a pseudocoelomate, and a coelomate is going to be things with a true internal body. So if I kind of draw this out for you, so if we're looking down upon that hollow ball of cells and we have the top of the gastrula is cut off. So I'm taking that hollow ball of cells and I'm cutting it like this and I'm looking down into it. I'm going to have a couple of different things. If I have a true coelomate, I have a tube within a tube body plan. Okay? This becomes your ectoderm. This becomes your endoderm. And this becomes your mesoderm. Okay? But what we find is as that pinches in, you have these various different ways of things dividing. So an acoelomate is not going to have a fluid-filled cavity. So it's going to be normally a more flat organism, might have a mouth down here, and then it's going to have some organ systems that kind of branch out right along this surface. So you really don't have a fluid-filled cavity. And that is an acoelomate. And an example of that is a flatworm. As you look at a pseudocoelomate, what happens is you end up with a little bit more of a round body cavity but it actually kind of pinches up in here like this. So you don't have that full internal space. And this is a pseudo. And you might have a branch that comes down here, but it's not really a full internal cavity. And your mesoderm is actually connected to your endoderm, so it's not completely separate. And this tends to happen in roundworms. That's one of the reasons why some of you that looked at those underneath the microscope could see right through the organism, and you could see all of those cavities. And the last one you have is a true ceolomate. And that basically means you have a tube within a tube body plan. And this starts with things like annelids, which are earthworms, and basically branches all up to other organisms other than echinoderms. And you'll need to take a look at echinoderms and decide what they are. So that's the essential of how you divide those up and how you determine what organism has what type of internal. Next thing we have is animal protection and support. And basically, the diversity of body cavities results in a different diversity of animals. And if you remembered on the video, we actually looked at the the skeleton, and it became from the ectoderm. So in some invertebrates, that ectoderm basically forms the outer skeleton, which is an exoskeleton. And normally these exoskeletons are actually made of this little term, which we noticed in fungi, which is called chitin. And then you also have the endoskeleton in animals that are vertebrates. I think you guys know most of that. It provides protection and support. And then you have the difference between invertebrates and invertebrates. And that is pretty much it for this. You're going to look more in depth into things like how the organism feeds, so you're going to describe that. You're going to look at how does it breathe, so you're going to go through all those individual organ systems that that organism has and really describe that. And I think most of those you're going to know, and it's going to be fairly easy in terms of that.